Okay, everyone. My name is Ivana Parker, and I'm an assistant professor in the Department of Biomedical Engineering at the University of Florida. I'm excited to share with you all the work that our lab is doing using machine learning to diagnose bacterial vaginosis and exploring ethnic disparities in that diagnosis. So just to give you an overview of the lab and what we do, we use multi-omics data, bioinformatics, and systems biology to understand how chronic inflammation increases risk for HIV transmission. And this work was really motivated by a postdoc that I did as a Fulbright in South Africa, where we were able to investigate how inflammation plays a role in driving HIV risk. So specifically, I found that bacterial vaginosis is a driver of HIV risk, and bacterial vaginosis also increases preterm birth, cervical cancer, and pelvic inflammatory disease. It's actually the most common vaginal syndrome among women of reproductive age, and it frequently recurs due to a lack of effective therapeutic strategies. So it's also presents as a health disparity, disproportionately affecting women of color, with 3 times as many diagnoses for Black women in the US compared to white women. So you can see how that also increases the risk of disparity of the other adverse effects. So what is bacterial vaginosis? It's a shift from a lactobacilli dominant to an anaerobic dominant microbiome, and it can be symptomatic or asymptomatic. Symptoms can cause extreme discomfort. However, it can also be asymptomatic, where you are having this dysbiosis of the microbiome, but you're not having symptoms, but you will still have increased risk for the adverse effects that I mentioned before. So the way that this is diagnosed is syndromically using Amsel's criteria, which is vaginal discharge, clue cells, which are cells that look somewhat like this, the presence of them, a whiff test, and a pH that's greater than 4.5. What is also used is Nugent scoring. So if you have a syndromic diagnosis that's negative, but you have a Nugent score that indicates BV, that's what we would diagnose as an asymptomatic case. So the way the Nugent scoring works is based on just identifying morph types of healthy versus pathogenic bacteria within the vagina, and it assigns a score based on the presence. So normal vaginal flora, you get a score of 0 to 3. Altered or intermediate vaginal flora, you have a score from 4 to 6, and then for BV positive is 7 to 10. And these are the scores that we've been using kind of as our base truth, as you'll see as we go along. Due to the advent of high throughput sequencing, this actually allows us to really determine the microbial makeup of the vaginal microbiome in health and disease. So the vaginal microbiome, as I'm sure you've heard of all different types of microbiomes, they vary very much individually and based on group, can also vary based on lifestyle factors, social determinants of health, and many other factors that we're trying to understand how that influences or increases risk for disease. So again, we have all these millions and billions of bacteria, up to 600 species in the vaginal microbiome. And so you can see how AI can be used to help us to understand and identify microbial communities of interest that may vary between groups or vary based on socioeconomic factors. So the research questions that we are trying to answer within my lab, and specifically that I'm going to present for this presentation, is looking at the complexity of the vaginal microbiome. Can we accurately predict BV using sequencing data, and does this hold true across ethnic groups? Is it fair? How do different model architectures compare in their ability to fairly predict BV across different ethnic groups? And can we also identify key bacteria that are important to consider as we look to identify better therapeutic targets for a personalized medicine? So for our first data set, we use this asymptomatic BV data set that was published by Ravel, and it includes the vaginal species composition of 394 women. So it also included the pH values and Nugent scores of these women. And one thing that was really helpful for this data set is that they also included self-identified ethnicity and had labels for white, Black, Asian, and Hispanic. And this data set has also been used by other groups for machine learning, but has not looked at how the algorithms perform based on race and ethnicity. And this here just shows the makeup of the data set and how there are these community state types, which are comprised of a dominant bacterial group. And so it kind of shows just the variation of types of vaginal microbiomes that were actually present within each group. So we trained 4 models, logistic regression, random forest, simple vector machine, and multi-layer perceptron, and we evaluated model performance across each ethnic group. Here, what I'll highlight is balanced accuracy, and then a false positive rate and false negative rate, because these allow you to see kind of what the implications of this incorrect diagnosis is. We also looked at a few other parameters that I won't present right now. So if you see here, we have balanced accuracy here on our y-axis and ethnicity on the x-axis. We have the different logistic regression here in red, multi-layer perceptron, random forest, and SVM in purple. You can see that overall there is a disparity in diagnosis. Overall, if you look at the performance for the total group, the performance is not bad, right? SVM tended to perform the best for all, for the total group. But if you look at it based on ethnicity, we see poorer performance for the Asian group, best performance for our white subgroup, and then in between for the Black and Hispanic group. If you see how this would translate into false positive rate or false negative rates, so we see that false positive rates, you have a higher diagnosis of positive when it's not really positive for our Black and Hispanic subgroups, and the opportunity for misdiagnosis is highest in our Asian subgroup here for the false negative rate. So it's missing a diagnosis for BV. So what we wanted to do next is run feature selection to see if we could improve the performance of these models. But also what was exciting for me to do with this feature selection is because what our lab does primarily wet lab work. We've gotten into the ML space. So for me, being able to identify bacteria that are important to study in different ethnic groups that are traditionally understudied was one of the hopes to be able to generate from this experiment. And so we do see this. So I've kind of, I've highlighted pathogenic bacteria in red, and then what would be traditionally known as beneficial bacteria in green. And you can look to see that with the feature selection, there are some that are important for all groups, or were very statistically significant for all groups for accurate diagnosis. But then you can see that it does vary across groups, right? So if you look at Megasphaera, it's very important for all groups, more important for white and Black, not as important for Hispanic and Asian. And then if we look here, like at atopobiome, we have all ethnic groups here. Was important as an important feature, but we see that it was mostly important, more important for the Black and Hispanic groups, not so much for the white subgroup, and somewhat for the Asian group. So here, this allows us to kind of see, again, what is traditionally studied in this space are Gardnerellas, our L. crispatus, as like this is the main pathogenic or main beneficial bacteria, but there are understudied bacteria that we are excited to take into our lab and actually understand how these contribute to inflammation and downstream HIV risk. So what we wanted to do next is to look at different subsets of BV. So this first subset was an asymptomatic cohort, right? So will we see the same disparities in different subsets of BV? So we have asymptomatic. Here we want to look at a symptomatic cohort. So if patients are presenting with symptoms, will the etiology change a bit? Will we be able to predict accurately? And down the line, we've also looked at cohorts of HIV-positive women as well. So this data set, we used a symptomatic BV cohort that also included ethnic classification. And this was a smaller cohort, 211 women. And the ethnicity classification terms included white, Black, Asian, Native Hawaiian, and Pacific Islander, or mixed. Because due to the small numbers, we created new categories of white and Black, and then we put the other groups together just for power for the experiments. We do understand that lack of data is also an important thing to consider in moving forward and trying to have accurate diagnosis for these algorithms. So this shows the breakdown of negative and positive cases by race and ethnicity. We did a Tisney plot just to kind of visualize our data, and you can see that the positive cases kind of cluster here, and then the negative cases cluster here, but then there's this interesting cluster here where there's a mix of both positive and negative, And you could imagine that would be a place where it'd be hard to accurately diagnose these participants. So performance was comparable across the models. I mentioned before that for the asymptomatic data set, the SVM model performed overall the best, but here we see random forest and logistic regression were comparable for when predicting using all of the data. But we do still see this disparity in model performance. If we look at balanced accuracy, we see that there was a lower performance generally between the white and black group, with other sometimes performing better than black and sometimes performing worse. And so we still, again, see this disparity. We tried to mitigate some of the disparity by doing some ethnicity-specific training and feature selection. And what we found is that this generally degraded model performance, which we believe also is due to the lack of sample size. So this leaves space for us to do more experimentation and to improve. This is another area that we're looking into. So here we came to feature selection, looked at different feature selection processes. And so what we found is that Gini index did improve performance for accuracy and diagnosis as it relates to balanced accuracy for our black subgroup. And again, moving on to the feature selection and why, again, it's important for us having the shared dry and wet lab is that we can then look to see which bacteria are important across the groups and start to explore how they affect chronic inflammation. So here we see the same Bacterial Vaginosis, which was important for all ethnicities, but you see it was very, very important for the white ethnicity, somewhat important for the group of ethnicities lumped into other, and then not so important for black. BVAB1 was very important for a black subgroup, but not important for any of the others. And you can see this trend as it continues. So I won't go through all the bacteria, but there are different ones that are highlighted that are lighting up for us that we can then go into the lab and kind of explore how these interact with the epithelial cells in the vaginal wall to promote inflammation. And so even if you look at the beneficial, the ones that are generally considered beneficial, such as an L. crispatus, you see also that these lactobacilli species are differentially important when using them to predict accuracy for bacterial vaginosis diagnosis. So in conclusion, the complexity is important to consider. A model may generally perform well, but the demographic information allows us to delineate differences in the performance between groups. Feature selection here has allowed us to form novel hypotheses for microbiome communities and that we are able to take into the lab and really explore in more depth. And I think this is really important to alleviate health disparities and also just to come up with better therapeutic interventions across the board for bacterial vaginosis. As I stated, that it generally recurs. And yes, I believe some of it is that the therapeutic targets just need to be better explored. So that is my talk. I would like to thank the team. The first work was published in NPJ Digital Medicine. The second part of it is in submission now. And I have some grad students and computational collaborators at UF as well that have helped a lot with this work.
Any questions?
Hello, thank you. I have a question relates to the feature importances across the models. Did the feature importances, so you mentioned Gini feature importances, did it line up with the feature importances derived from like the random forest or the FSBM or the logistic regression coefficients?
No, that's a great question. So what we ended up doing is we took our best performing model and we did the feature selection just based on that one. So I believe it was random forest for this last for the symptomatic data set, and we just went there. We are doing more testing to understand how those coefficients align with the feature selection as well, and looking at different ways to measure feature importance.
Thank you.
You're welcome.
Are there any other questions?
Hi, first of all, I really like your approach towards it, and I really like how you differentiate the ethnic groups of each one, and you found the different genes on each one. My question to you is, do you guys saw or take into account any of the age difference in between these? And do you think it makes a big difference, or could it balance the gene?
That's a great question. So we do not take into account age in this study because that information was not available for these data sets. We are very interested in age as well as menopausal changes and just a lot of different lifestyle factors that do affect the vaginal microbiome. And we are working to kind of pull together other data sets that may have that information, metadata as well, but we're also building those cohorts within our lab because some of them just don't exist. And that is what we're doing. So I do think that is really important, and we're looking to be able to incorporate that into future analyses.
Any last questions? All right, let's thank our speaker again.